Okay, this video is more about proteins or amino acid sequences. The protein is made out of amino acids, and there are 20 amino acids. You attach those amino acids together, and they fold up into a protein. But the three-dimensional shape often relates to the chemical abilities of that protein, what that protein can do within the cell. And the sequence, though, the sequence of the amino acids and how they fold up into the protein basically give it its chemical nature. All right, but we can analyze the sequence, each amino acid attached to the adjacent on either side of each amino acid, unless you're at the ends, will be another amino acid. So it's a linear sequence, and each amino acid has a different chemical property and different chemical functions. And so the idea is, by analyzing that sequence, you can tell perhaps what the protein does. And if you change those, all right, you change the chemical properties of the protein. So we're going to go through here and kind of look and kind of get some explanations on how that works and how we analyze that. So in molecular genetics, you have homologs, orthologs, and paralogs. Now, we did a worksheet on homology. Now, homology, it's two sequences with similarity. Homologs usually represent genes that are related to a second gene by descent. So the idea here is homology is measuring or kind of looking at the two sequences with the assumption that, hey, they are descendant from a common ancestor. Very similar to you have an ancestor, you have a common ancestor with a Neanderthal, which would have been maybe some type of ape from millions of years ago, all right? And just as you and your sibling have a common ancestor in one of your parents. Well, the whole idea is you go far enough back and you have common ancestors with various organisms. So something like you and a fly have some common ancestor way, way back, some creature that both flies and humans evolved from. So the idea is these homologs this common ancestor idea doesn't just relate to individuals, but essentially it relates to the sequences in their genes, and perhaps you can find those similarities, remnants of similarity, which lead you to believe that you have a common ancestor. So that's the idea of a homolog and why we look at it. Now, orthologs are genes from different species that evolved from a common ancestral gene. So it's kind of the idea, orthologs, homologs. This one is across species. This is kind of within the organism. I guess I kind of misspoke, but homologs, genes are related to a second gene by descent from a common ancestral DNA sequence. So the idea is this is a sequence, and this is the broader one. This is the more specific one, if that makes sense to you. This is the sequence related to another sequence. This one is the sequence in a species related to the sequence from another species. It's kind of like they commonly evolved. I kind of misspoke when I said what a homolog was. Better definition for that, the way I described it, would have been an ortholog. But homologs homologs can be within the same organism. In other words, you can have your genes copied within the organism. All right. Paralogs, exactly, that's where the next one is, are duplicated genes within a genome. So the whole idea is homologs are the more general one, and then you can get specific types here, duplication and across species, duplication within the genome, and then across different genomes in different organisms. So it's kind of difficult to kind of get the idea, but homologs are kind of the more general classification, and then these are specific cases, if you want to think of it that way. Actually, this picture, you're right, does a much better job. See, all of the orthologs and paralogs are homologs. All right, that's kind of the idea, is what I was trying to get across. and these are like different genes. The alpha globin, beta globin, they're both the same. They're different versions of the same protein or gene, if you want to look at it that way. But they have slightly different function, slightly different shape. But we know that they came from the same ancestor, kind of thing, way back. And so alpha and beta are paralogs. Orthologs are across species. And so these beta globin genes, there's a mouse one, a chick one, and a frog one, and a mouse, chicken, frog here. But really, these two, at some earlier point in time, were duplicated in some organism. In other words, once there wasn't an alpha and a beta. There was just the gene, the globin gene. And then at some point in some early ancestor, before there were mice, chicks, and frogs, there was a duplication. So the globin gene was copied. And gradually those two mutated a little and changed from one another to become alpha and beta. Now, as time went on, frogs, chicks, and mice evolved. And when that occurred, then they each had their own version of the alpha and beta. And they evolved, and they're all slightly different. And so you can compare all these sequences, and this is all because of gene duplication. You kind of go back, and this is kind of going backwards, and they're trying to find the early globin gene. What do all these have in common? And really, all this will really tell you is what pieces didn't change over evolutionary time. And that actually is important, because the pieces that didn't change across any of these, those are the ones that are required for it to have its correct function. So if you were to take the sequence for each of these 6, and you were to, well, remember, these might have a slightly different function than these 3. So I should be careful what I say here. There's probably some pieces of the sequence that are required to be a globin, whether or not you're alpha or beta. And those pieces would never change. Then there are some pieces that are required for you to be an alpha, and those pieces never change over here. And then there would be requirements for you to be a beta, and those sequences would be required in each chick, frog, and mouse sequence would be required to be there for it to be a beta. And the other things could change, but whatever things are in all 3 of these are probably required. All things that are in all of these are also probably required. And then there will be a subset that's required for all 6, and that's what's required to be a globin gene. Not an alpha globin or a beta globin, but just a globin. And when you find out what's required in all 6, you kind of know, like, okay, this is the most important piece for the function of it. Then you can find out what the most important piece is for being an alpha, and the most important piece for being a beta. And then you can go look at things like what's different between the frogs, the chicks, and the mice, and things like that. But generally, that's how we look at these things. All right. There are many. This is giving you an example. Drosophila has many homologs of the following mammalian proteins or peptides. In other words, we have a copy of this gene, and flies have a copy of this gene. This is for myosin, or heat shock protein 70, which is a chaperone. It helps other proteins fold. This is the suicide gene, the one that if you get cancer, this gene says, okay, time for you to die. You're kind of confused cell. You need to essentially lyse. And if this one gets mutated, then the cell can't kill itself, and therefore it often becomes a tumor. This is the beta globin and insulin gene. So both flies and humans have all of these genes. Now the sequence that produces them is slightly different, and they don't work quite the same, but as a general rule, they do have the same function in general, like insulin, to help regulate glucose in the bloodstream. Both the human and the fly gene will have the same function, all right? But they'll be slightly different, and because the fly's biology is slightly different than the human biology. There have been mutations in other places that cause the fly to be different from a human, obviously, and the insulin gene had to adjust to those, and so the protein is different, but it still has to have certain key features for it to be insulin. And by comparing humans and flies, you can look to see where the important parts are, the parts that are required to make it insulin. And you can't change those, or you get some pretty wild mutations, kind of like the ones, you know, your eyes disappear, your wings change shape if it's something in a fly, something like that. But by comparing humans and flies, you can get an idea. If something is the same in both a human and a fly, then it's pretty important. You don't want to change that piece of that part of the protein, and that means you don't want to change, of course, the DNA, which produces that part of the protein. All right, we keep going. Now, realize, of course, that amino acids have 20 letters, not 4. This particular beta-globin amino acid sequence for this gene had 147 amino acids. All right, and so it always starts with methionine, if you didn't know that. The first sequence, AUG, is the codon for producing methionine, and it's always the first one here. So let's move on. The human is quite a bit different. It has 458 amino acids. So you're going, well, those don't look like the same gene to me. The amino acid sequences are wildly different. MVH, MSI, that's not the same, but they have the same function, just in different organisms. There will be places that they are similar, and that should be evident if you were to align or compare these two, like run it through BLAST or dot plot, some of the other programs we've seen. But there is a motif. That's what we call the little pieces that don't change. Conserved region, 10 to 20 amino acids that are required for it to function and be an insulin gene. That is enolase. I'm sorry, enolase is what they're looking at. So let's find out what that piece is, and it's right here, GTW. Now there are some differences even here, but you can see this region does not change, and that's the key. Oh, I'm sorry, we've moved on to a different organism. I'm sorry. This is a lipoprotein. They're comparing it between three different bacteria. I wish I'd stick to the same, the same sequences. All right. So just remember the proteins fold, and they have a three-dimensional structure, and how they fold up is dependent on the individual amino acid sequences. And so these parts can change, not as important. But if there is a piece that has some specific chemical function, often you can pick that out, and that will, every protein that has that function will typically have, say, a signature, or what we call a motif, that determines what it does. All right. Now this is a type of a diagram that shows you the importance. So the larger the letter, the importance. So there really needs to be an R here, or there needs to be somewhat important to have an L here and an F, maybe an L here. Medium low importance here, but very important for a P here. Medium for maybe an E, high importance for a T. It's because we created a consensus here. We look at all of these and they go like, all right, these all do the same thing. Here's the piece that's pretty similar between all of them. Notice you always get a G, 2 out of 3 you get a T, you always get a W, always get a Y, that sort of thing. And that's what they're kind of looking at here. I don't think this one goes with this one, though. I don't know. D, K, K. No, I don't think this one goes with... I wish they would keep it consistent. All right, so there are searching for protein motifs, ProCite, PhyBlast, Meme. All of these are different. We've done BLAST. This is the one that looks for patterns in proteins in a specific way, and it looks in the surrounding alignment, local alignment. All right, domains are just regions that tend to have similar properties, similar shapes. So you might have a domain within a protein, and you might look for domains, and we kind of know which amino acids, when they put together in a certain sequence, get the same domain. So you might get mutations that alter the thing, and it doesn't change because the shape doesn't change. And if we see certain patterns, we know that, oh, okay, it has the right shape in general, even though we can't tell here. I can't tell that these are similar in this region, but if they all folded up and had the same shape, then okay, that's fine. The shape is the same, and then the chemical function's the same. So yeah, it ends up being the same function, the same protein. In other words, it does the same thing. Let's see. So these domains may give you shape, while others are found in protein with certain function, but shape and chemical, I really should say chemical activity. So the shape is important, all right, which is that's related to chemical activity as well. But then there will be certain chemical domains. They really should distinguish here. There are chemistry-based domains, and then there are shape or structural-based domains. Now, the shape, of course, is related to chemistry, and the chemistry, of course, there's shape involved there, so they're not quite the same, but there's like this overall shape, and then there's like local chemistry, and there's more than one type of domain that they're kind of implying here, but not actually saying it in so many words. All right, so here is a case of a protein where we understand the shape, but you also have to have certain letters, certain properties within that shape. That's kind of the chemistry part, because you have a zinc that binds to the protein, and so you have to have two H's, two C's over here for the zinc to fit in here. So you can't change that. You can change maybe these and get the same shape, but you have to have these particular residues in this particular order to get the zinc to go in. So the shape is important, but also you may need specific residues to get this zinc to fit in there. So that's more of a chemistry focus. And this is called a Zinc-Finger, by the way, which it's something that makes this a DNA binding protein. This would be a protein that would bind and stick to the DNA. And you can see here we have they've highlighted the ones that never change. You go through 9 different versions of this protein from different organisms, and look, C never changes, right? You see that? Then there's another C here. That's the other one. Then the H's. Some of these others can change or move a little bit, and it's okay. But as a general rule, you can't change these. They have to be in a particular order, and that's so that it can stick to the DNA. These would be the pieces that, or I'm sorry, in this case, these are the pieces that stick to the zinc. The DNA fits in between the tubes and curly things here are the structure of the protein, and it fits around this DNA strand, and it fits right into the major groove, I believe. In DNA, there's a major and minor groove here, and the zinc fingers fit into the particular shape of the DNA, and if that shape of the DNA is just right, this protein will fit in there and stick to the DNA. All right. Usually you can determine if it has a domain. It could be a long domain, the length of a protein. It could be a subset of the sequence that could be located at different places. It could be repeated. All of these, you might look for these domains. These are just common sequences. This would be like a domain, a motif for the Zinc-Finger. All right, and so maybe you need a Zinc-Finger is closer to probably something like this. A Zinc-Finger would be one of these. It's a subset of the whole protein. So you might have a Zinc-Finger here. You might have it in another protein in another organism. It might be way down here, but it's going to have a Zinc-Finger to give it the right shape to attach to the DNA. Are proteins that share only a domain homologous? Well, yes, to some extent. It's a relative question, and yes, that's what they say here. Depends on the function, the mutant phenotype. So there are a lot of factors, but at least the regions are homologous. So the MBD domain or motif is located in each of these, and it provides at least a similar function, and so we would call at least this region homologous. Mutations are changes in the genetic material of a cell or virus. We know that. Point mutations are just one base pair change. So that one base pair, if it happened to cause a change in the amino acid sequence, there's a potential that it could change the function of the protein, and therefore you get a mutant. It could change and not change the amino acid sequence, and you wouldn't get a change in function. It could change and change the amino acid sequence, but still not change the function. It still wouldn't be a mutant. So it's only considered like a major mutation, or you would call it a mutant in the sense of the overall fly. It has a mutant. It's a mutant if you get some type of phenotypic change. And just because there is a mutation in the DNA doesn't necessarily, you can't necessarily say that if it doesn't cause a phenotypic change, we generally don't pay any attention to it, actually, is what happens. It technically is a mutation, but it just might not be an important one whatsoever. And you would never study it, probably, and never look at it, and it's not important. You have to figure out if these mutations are important or not, whether or not they altered the amino acid sequence, typically is what we consider an important change. So here we go, and that's what this diagram is kind of pointing out. We have CTT, and on the other strand GAA, and what it did was our mRNA is here, and it says this should be I guess that's glutamic acid here as the amino acid sequence. Well, we get a one-letter change, all right, A to T here, and that now codes for a valine. So therefore, you get sickle cell anemia. So you had one nucleotide change, which altered this particular codon, which therefore caused the protein sequence to be different. It was supposed to be glutamic acid, glutamine, don't know, glutamine. All right, but over here it's valine, and valine is the wrong one. And so if you get valine here, basically the hemoglobin actually is the wrong shape. That's actually what it is. Actually, it's not the wrong shape all the time. It's only a wrong shape with the wrong pH. Different pH, the protein folds differently, and it causes sickle cell anemia. So when here, it stays the same shape regardless of what's going on inside the bloodstream. But in this one, if the pH changes, it's actually the amount of sugar in your bloodstream changes, which alters the pH. Then certain things occur, and the hemoglobin is altered, and therefore causes the actual cell to change shape as well. Actually, it changes shape and becomes, I guess you could say, the blood cells are, well, the protein itself sticks to itself, and so they clump, and when you clump, it alters the shape of the cell. You alter the shape of the cell, and it can't move through the bloodstream as easily, and you get clots. All right, types of mutations. These are point mutations. That means just one letter change. You could have a substitution, insertions, or deletions. And as I mentioned, they could be silent, not do anything. It could be disabling, where it makes you're unlikely to function as well. Or it could be lethal. The cell doesn't even survive past maybe one cell or a few cells, and then they all die before birth even. So if you're talking about something that is more than one cell, like it happened with bacteria as well. But you could have different types of point mutations, and they could be any one of these. And you could have substitutions. A substitution is when you go from an A, say, to a T. You substitute one for another. But you can also have insertions and deletions, and these, of course, as we saw in the worksheets, are the more important ones because you change what's called the reading frame. And here is an example of the reading frame being changed. No, we're not changing the reading frame here. We're just substituting in this first one. So they substituted an A instead of a G, and of course, what did that do? Well, in this particular case, it didn't change the amino acid because both of these codons, the GGC produced glycine, and GGU also produces glycine, so this is one that would have no effect whatsoever because it's silent, because the codon here, there's more than one codon for glycine. Now here, you did get it if you changed the letter over here. Now you end up with serine instead of glycine, and of course that might change. It's called a missense mutation. Now a nonsense mutation is when there's no, you didn't just change, it's a more important mutation. You didn't just change the one amino acid for the other, but in this particular case you changed which amino acids were there, or in this particular case you created a stop codon where the amino acids are not produced at all. So in this particular case, by changing this to a U, you now have a UAG. Here UAA was where the stop codon used to be, now it's up here. So this protein, you cut off what? You cut off 3 amino acids from the end of this protein. Now a frame shift is probably the worst in the case of you take one out or you add one. If you add one, you might also, in this case it caused a frame shift for a nonsense. So it's a frame shift is when you change how it's read. In other words, you read these like 3 at a time. So it's 1 2 3 1 2 3 1 2 3. But if you take one out, now you're not reading the same 3. You're reading 1 2 3, and it's not just changing these 3 anymore. I could change this and I alter just this one. But when you get a frame shift, you're shifting all of them down. So now you're not reading the same 3. Instead of reading these 3, these 3, these 3, with a frame shift, you're reading like a different set of 3. You're starting here and reading these 3, these 3, these 3. So after a frame shift, you are probably creating completely different set of amino acids on the other on the right hand side of the frame shift where it begins. And so all the amino acids coming along are all going to be different, and you're certainly going to get a different structure, if not different function, from that protein. So those are more likely to cause a major change in your organism, all right, if you get a frame shift, than just the points, because you can have, like in this first one, where you could you get a change, but it didn't change the amino acid sequence. But down here, this causes major changes, these nonsense mutations and these frame shift mutations. Just missing one can also cause a frame shift. So everything after the frame shift from missing one, so adding or deleting one causes a frame shift. And so everything afterward is going to be different. This one eliminated the stop codon. So say this one, and say it changed leucine and alanine, but used to be stopped, so that was where we would stop making the protein, but now it's going to keep on going. And of course it keeps on going, you're going to get all kinds of things added at the end. Here you're missing an amino acid, you just took out 3, and so you can have a deletion that doesn't cause a frame shift if the deletion is in multiples of 3. So here we altered it and you took out 3. Well, that just means you're going to make this without the lysine. All right, now substitutions are just changing the sequence a little. You change from an A to a T, and often they are silent. They have no effect. Missense, they still code for amino acid, but not necessarily the right amino acid. If this is not in an important region, might not do anything. And nonsense can change the amino acid codon into a stop codon. So if you do that, you're almost always going to lead to something. If that last part was important at all, you know, it's not going to be there. So of course that's going to change what's happening with that protein. Of course, insertions and deletions other than multiples of 3 cause frame shifts, and there are almost always, like I said, disastrous effects, and just keep that in mind. The frame shift is the bad one, though nonsense is also bad. You can have other things that can cause just as bad a problem. But the ones where you just have a single base change, usually not a big deal. There are lots of those. For a single base pair substitution to be bad, it has to be in one of those regions that's critically important to the function of the protein that we've talked about. Let's see, transposons, transposable elements. Well, these are jumping genes, and they can jump inside the gene. They're like large pieces of DNA that jump around the genome. And if they happen to jump in or near a gene, then they can alter, if not how that gene works, they alter how it's regulated, perhaps. And so a transposable element can definitely cause problems. All right, now Barbara McClintock she discovered them. She found them in corn. She was studying the coloration of corn and realized it was caused by a transposon, a piece of DNA that jumps around. And in some of the kernels, it jumped here and it jumped there. And when it jumped, it caused the patterns to change. And she discovered it. And at first, everybody said, well, that's kind of cool. No big deal. Who cares that much about corn? Actually, corn's important, but you know, generally, they didn't think this occurred in every organism. Now we know it definitely occurs in every organism, and transposable elements are probably one of the most important discoveries for evolutionary biology. Most of the regulation of your genes is probably derived from transposable elements. Well, that's speculation, I should say, on my part, but that's the general consensus coming forward, is that transposable elements are far more important than we ever knew. There are different types of transposons. Some have a DNA intermediate, and others go through an RNA intermediate. But these are just sequences that kind of, during replication, usually they get copied and they end up moving into another part of the genome. They can either make copies, or they can cut and paste. They can cut a piece out and that piece move, or you can have it so that they make copies and those copies go out and insert themselves all over the place. This is just the idea here. It's a transposon. This one is a copied one, and then it would insert in here. And of course, if there was a gene here, man, you just cut this and splice this piece in the middle of it. That's going to, of course, disrupt things. Now this one is an RNA version of the same thing. Multiple copies, they move around. In primates, the main one that we talk about is the Allele element, and I study Allele elements. And the Allele is likely the primary driver of the evolution of humans from apes to humans. There's a certain Allele that tends to jump into the promoter regions of genes that affect the nervous system, and those are probably the ones that allowed us to get more variety, I should say, in those genes, and allowed us to expand the size of our brain and the functions. So they're important in evolution. Many types of retrotransposons. There are different types: lines, signs, L1, L2. There are different versions of this that's kind of beyond this class, but just realize that they exist. Long terminal repeats. How basically you get these little repeats repeating many times next to each other, and you can expand the genome. The genome expands; it gets bigger when you get a lot of these things copied, and that's important. It's kind of like extra space to make more genes, I guess. The human genome is expanded and has a lot of these Allele elements and other elements like these LTRs all over the place. Now in Drosophila, they have a different set than we do. They have this one gypsy here, for instance. All right, so you might want to read some of these. Ask me questions. Barbara McClintock is becoming pretty famous, mainly because she was kind of ignored at first, and people didn't think there was much to it. And as time goes on, more and more we realize just that was pretty central. It was important that she discovered these. The jumping genes are really important to the to just how everything works. So especially disease. So Barbara McClintock is now pretty famous. There are a couple of books written about her. All right, I actually worked with a professor who was in the same lab with her, at least for a few months, and knew her quite well. They went to graduate school together, and she's a very nice lady.